Well, hello everyone, and thank you for joining us today as we have one-on-one discussions with our scientists within the MIAMI Project to Cure Paralysis. My name is Dalton Dietrich, and today I have the pleasure of talking to Dr. Masumi Ghosh, who is Assistant Professor in the Department of Neurological Surgery and a very important member of the MIAMI Project to Cure Paralysis. Hi, Masumi, how are you doing today?
I'm doing good. Thank you, Dr. Dietrich, and thanks for inviting me. It's my pleasure to discuss my work.
Fantastic. So yes, we've been looking forward to talking to you about your research efforts that are not only at the University of Miami, but also at the Miami VA, which we can talk about later. But your research, as I understand it, is concentrating on neuroinflammation and the innate immune response to injury. We know that inflammation can be bad, but it can also be good. And a lot of your work is talking about how inflammation can actually help repair the nervous system after injury, and focusing on the microglia, the microglial cell in the central nervous system. So can you tell us about the microglial cell? Why is it important, and why is it a target for potential therapeutic interventions?
Yes. So microglia are the resident macrophages of the central nervous system, and they are a component of the innate immune system. So under normal circumstances, it plays a very crucial role in the development of the central nervous system. It's important in neurogenesis, in shaping of the neural circuitry. It's an important phagocyte, helps in clearing the debris, fights infections in the central nervous system, if any, and also produces growth hormones or growth factors, as well as are important in sustaining of the blood-brain barrier. But following an injury to the central nervous system, or even in the condition of a neurodegenerative conditions such as ALS, Alzheimer's, multiple sclerosis, Parkinson's, stroke, etc., it takes up a pathogenic role. So microglia, they are really unique and versatile, not only just morphologically, but also functionally. So after an injury to the central nervous system, what happens is they take up a pathogenic role. And by that, what means is that as much as they help in clearing up the debris of the dead and damaged tissue cells, they also end up, they become very activated, and they end up producing an array of pro-inflammatory mediators. These mediators, examples of some of them are like TNF-alpha and IL-1 beta, which are pro-inflammatory cytokines. There's ATP, excitatory molecules like glutamate, various toxic proteases, as well as reactive species like reactive oxygen and nitrogen species. They're highly cytotoxic, and what they do is they end up triggering cell death of the neural tissue, neurons, oligodendrocytes. And this ultimately results in a substantial damage to the central nervous system, which is much more than what the initial injury causes. So this is the pro-inflammatory phenotype of microglial cells that causes inhibition of regenerative repair. It results in loss of function after spinal cord injury, and it is actually a potential initiator of the secondary tissue damage. Interestingly, after injury, it's not just a single population of microglial cells that are present in the environment, microenvironment of the injured spinal cord. It's actually a mix of multiple populations that vary in their states of activation. And if you really look at the timeline of microglia, which is normally activated immediately after spinal cord injury and remains activated throughout the entire acute, subacute, chronic stage, of course, the peaks may vary, peak of their activation. There are spikes of microglial population that come up that are not just inflammatory, they are neuroprotective or anti-inflammatory, which are useful. So what decides the activation state of microglia? It's actually the microenvironment, the signals that are obtained from these dying cells that triggers the pro-inflammatory phenotype, or sometimes there are cytokines that are present that are produced by astrocytes, say by neurons that could trigger an anti-inflammatory or even a neuroreparative phenotype. So based on what environment microglia is, it decides the activation state. Although largely it is the pro-inflammatory, undesirable, and cytotoxic phenotype in the injured spinal cord or injured brain after TBI, but still there is a way to promote or induce or even convert these cells to a neuroreparative phenotype using biochemical or pharmacological approaches.
Yes, so it's really interesting. You can look in materials from human studies that have undergone spinal cord injury or stroke or traumatic brain injury, and you're so right, years after the injury, these microglia just stand out as still being very active and potentially releasing pro-inflammatory cytokines. So it's a really interesting target. So you talked this pro-inflammatory or pro-reparative process. So what types of therapeutic interventions are you thinking about that could change this phenotype? These are such interesting cells and so plastic in terms of, you know, modifying the environment. But when you turn them on, sometimes it's hard to reverse that phenotype. So what types of therapeutic interventions are you thinking about these cells more pro-reparative?
Yes. So in our laboratory, we are using both biochemical and molecular approaches to trigger or induce the pro-reparative phenotype in microglia. So one of the approaches that we take is more like a combinatorial approach, and that involves one, Th2 cytokine, which is IL-4, interleukin-4. This cytokine basically is immunomodulatory and has anti-inflammatory properties. So not only in our lab, several other labs have used this cytokine. It has been administered systemically in experimental models of spinal cord injury in the acute phase. And there is an improvement in the functional restoration, to a significant improvement in the locomotor function in models of spinal cord injury. There's change of microglial phenotype observed, that they are more pushed or driven towards the pro-reparative phenotype. They have various characteristics that now do not match the inflammatory, but the anti-inflammatory phenotype, and they are in fact more neuroprotective because they produce, say, growth factors, and they help in at least in vitro tissue culture experiments, has prevented cell death, neuronal cell death, when used in co-culture. So in our laboratory, we use a combination of this anti-inflammatory cytokine, IL-4, along with molecules that elevate levels of cyclic AMP in cells. So cyclic AMP is another compound. It's a small molecule that ends up inhibiting the pro-inflammatory phenotype in microglia. We noticed both in vitro as well as when we administered this in subacute models of animal models of spinal cord injury, we noticed that these two compounds, they seem to act in concert, and they show a potent synergistic effect by converting or promoting a population of microglia within the spinal cord that is more not pro-inflammatory, but anti-inflammatory. And at the same time, we observe significant improvement in both gross and fine locomotor movements in animals with injured spinal cord. So this is something that I definitely see holds promise in the future in the clinics, and we hope to see that it will be used in human SCI at some point.
So in response to talking about human spinal cord injury, of course, many times targeting inflammation, we're talking about preserving tissue integrity. But in terms of some of the behavioral or clinical outcomes of spinal cord injury, one of the important quality of life issues for people living with spinal cord injury is neuropathic pain. So how are your studies potentially affecting the pathogenesis or the generation of neuropathic pain in patients living with spinal cord injury?
Yes, so microglia, they get activated after spinal cord injury above, at, and below the level of injury. So these pro-inflammatory or activated microglia, like I mentioned, they produce an array of these pro-inflammatory mediators, starting from pro-inflammatory cytokines, chemokines like MCP1, and they have all these reactive species, glutamate. All of these together collectively, they end up altering the hyperexcitability of the dorsal horn neurons, along with, of course, they end up sensitizing the nociceptive neurons. They end up blocking the inhibitory GABAergic and glycinergic pathways. They end up even resulting or triggering death of GABAergic neurons. So together, they result in the development of neuropathic pain. So what we are trying to do in our laboratory is use these combination of IL-4 and cyclic AMP in subacute models of spinal cord injury. We deliver them systemically, and then we see if there is a change, and if we can alter the phenotype of microglia and see if we can reverse or either the development or the persistence of pain. So this is a current project that's ongoing, and we already have exciting results where we see improvement in reduction of cutaneous allodynia with the use of IL-4. We are yet to do various combination molecules, but that's been very encouraging so far.
Very exciting. As I mentioned previously, you also have an appointment at the Miami VA. Can you tell us a little bit about how your research impacts veterans? Obviously, it's a very large group of individuals. And you talked about neuroprotection, but usually now we're talking about a chronically injured patient population. So how does your research touch that military care programs?
Yes. So now spinal cord injury as well as neuropathic pain, they are all, they remain, they're chronically, you see the activated microglia, you see neuroinflammation, and these are not just in the subacute, acute, subacute stage. It remains, inflammation is there in the chronic stage too. So we want to use this therapy of this anti-inflammatory cytokine IL-4 and these molecules that elevate cyclic AMP, but largely in regards to IL-4, there are some issues which may not show efficacy in chronic patients. And in veterans, we have a large number of SCI patients that have neuropathic pain. So treatment of these veterans is necessary, but we have to have an alternate approach so that we can see beneficial effects of these anti-inflammatory cytokines. So one of the approach that we have developed along with collaboration with Dr. Damien Pierce and Dr. Jackie Sagan in our department is we are trying to use not only just IL-4 cytokines subacutely, but for chronic. Of course, now we are doing it in experimental animals, and we want to deliver this cytokine, which unfortunately does not cross the blood-brain barrier. Neither IL-4 is produced in sufficient quantity within the injured spinal cord. So to reach the inflammatory cells or activated microglia so that it does not or reduces production or reduces trigger of neuropathic pain and inflammation, we need to have a local delivery of this cytokine within the injured spinal cord. There's also another issue. If we deliver this cytokine systemically, patients of spinal cord injury often have infections. So we do not want to use the cytokine, anti-inflammatory cytokine, and suppress the immune response. We need to have an active immune response present peripherally in the system. So to do that, we are using an alternate approach, which is combining cell therapy to deliver this anti-inflammatory cytokine at the site of injury. For this, we have used Schwann cells. So Schwann cells are the myelinating cells of the peripheral nervous system. And they are currently in the clinical trials, transplanting them in the SCI patients are undergoing clinical trials. We have genetically altered the Schwann cells using viral vectors so that they can secrete active forms of IL-4. Now, if we transplant these Schwann cells at the site of injury, then they will serve as an excellent delivery vehicle to locally deliver the cytokines right there where it's needed. And at the same time, we can bypass the problem of patients acquiring infection if it is given systematically. And also the problem of crossing the blood-brain barrier is not there, which normally gets sealed at the chronic stages. It's only open for a few days after injury and spinal cord injury. So currently the study is ongoing. We are in the process of using Schwann cells that deliver IL-4, anti-inflammatory cytokine, and we want to see if we can benefit from it, for changing the activated microglia or driving them more towards the anti-inflammatory phenotype to see if we can see a reduction both in neuroinflammation, chronic neuroinflammation, as well as neuropathic pain.
Very exciting, very exciting. Yes, the Schwann cell project continues to move forward. And as you know, we're now trying to isolate exosomes, these extra cellular vesicles that are taken out of the cell. So maybe that might be an alternative. Instead of having to transplant the cells, you could actually maybe induce the exosomes from the Schwann cells, or maybe the exosomes are already transduced through your methodologies, and if you use it systemically. So those type of things we'll think about in the future. So how has COVID-19 pandemic affected your program? Is it still very busy? Has there been limitations? Tell us about that.
Yes. So we had some ongoing in-life critical animal experiments, which started much before this COVID-19 situation happened. Since we are working with chronic animals, animals are already injured. We want to collect behavioral data. We have some very good, unfortunately, I was lucky to have some very good lab staff members who are coming in on alternate days, trying not to have overlapping stay in the lab. They're keeping social distancing, and they're doing an excellent job in acquisition of behavioral data after using all these compounds that we are using for reducing inflammation. Other than that, we have a good amount of data that we have already acquired. So both them and I are quite busy trying to analyze the data, write papers, and hopefully we'll get something out very soon from the data that we have.
Excellent. Yeah, we always talk about how this pandemic has really changed the way we're doing research, but the scientists are so dedicated, they're still trying to find ways to move the science forward. So thank you and congratulations on all your work, and thank you very much for meeting with us today, and we'll have you back to discuss some of your research programs in the future. Thank you.
Thank you so much, Dr. My pleasure.